Stratified squamous keratinized epithelium, palm of
Hmm.
hand,
Hmm.
pain hematoxylin and eosin.
Hmm.
One is stratified squamous keratinized epithelium.
Hmm.
Two, papillae.
Hmm.
Three, connective tissue with collagen fibers.
Hmm.
Four, excretory ducts of sweat glands.
Hmm.
Five, stratum corneum.
Hmm.
Six, stratum granulosum.
Hmm.
Seven, stratum spinosum.
Hmm.
Eight, stratum basal.
We are going to see the skin. Skin, we have first surface, then we have folds like this portion, and then we have this portion, and then we have adipose tissue here. Major portions of the skin, or this portion is known as epidermis. This portion is known as dermis, and this portion is known as subcutaneous tissue. Papillary dermis and deep dermis. What do you call the thing papillary dermis? Because they are like papillae going into the epidermis, and you can see some nice vascular, I have to draw it from red color, vascular plexus in between the papillary dermis and the deep dermis, and in between the dermis and the subcutaneous tissue. Now let us go to this portion and see how they are thrown into folds. These are known as rete ridges, and normally it is there. For example, there is a skin and it is seen like this, and the adipose tissue is there. It means atrophy. In atrophy, the rete ridges, for example, scleroderma, rete ridges will be gone. We will, and they will have some adnexal structures. Okay, this I have not drawn the adnexal structures because I want to understand there are mainly three portions: epidermis, dermis, and subcutaneous tissue. So this is the surface and this is the basal. Let us see what are the types of cells in the skin. There is one layer here. So these are more like round cells or cuboidal cells, and they can show some mitosis. And I will put nucleus to this. Since these cells are dark, I am not showing the clear nuclei. So this is the basal layer, okay, sitting on the basement membrane. So this is the stratum basal. Above these, some cells will become mature, they will become larger, and they occupy most of the epithelial thickness or epidermal thickness. And these cells, when you see, they will have spinous processes. To draw the, I will draw the nucleus in the cells. This spinous process I am not going to draw for all these cells. They will have spinous processes. So this is what do you call? What do you call, ma?
Stratum spinosum.
And if you go above, there are some cells that will have more flattened, more maturing. They will have a nucleus, and they will have blue or pink granules in them. So what is this layer, ma?
Stratum granulosum.
Go above, there will be some very flattened cells. They won't have that many nuclei and they will be full of keratin. Stratum corneum. Now what are the layers? Tell me, ma.
So there is
Veda.
so basically skin is divided into epidermis, dermis, and subcutaneous tissue.
Very good.
And in the epidermis, there are further some layers. Starting from the base, there is stratum basal.
Very
Over
good.
it, there are cells with spine, like projections, so stratum spinosum. Over them, there will be cells occupied with granules, so stratum granulosum. Over that, stratum corneum.
So this is a normal skin like our forearm, but what happens in the palm? There is a difference, isn't it? So there will be hyperkeratosis. So that is the keratinized layer. So by looking at the amount of keratin, you can differentiate sole and palm from other structures in our body. Those are all the two areas. And this is like nice basket weaving. This is known as basket weaving pattern, which we should not call it as hyperkeratosis, because these are all normally hyperkeratinized area. If you can see the palm, you can see it is different from the dorsal aspect of the hand. These are all the hyperkeratotic area, which is a normal component. This is the understanding of the skin. And what is this portion, ma?
Papillary dermis.
Super. So this is papillary dermis. This is deep dermis, or
Deep
otherwise
dermis.
known as reticular dermis,
Reticular
because there
dermis.
will be a lot of reticular fibers there. Okay. So it is also known as reticular dermis, and beneath that,
Subcutaneous tissue.
finish
So,
reading again.
Stratified squamous keratinized epithelium, palm of hand.
Hmm.
One is stratified squamous keratinized epithelium. Two is papillae.
That is papillary dermis.
Papillary.
Hmm.
Three is connective tissue with collagen fibers.
Reticular dermis. Hmm.
Four is excretory ducts of sweat glands.
So adnexal structures we will see when we are seeing the skin. Hmm.
Five is stratum corneum.
Super.
Six is stratum granulosum.
Very good.
Seven is stratum spinosum.
Very good.
8 is stratum basale.
Abhijit, read the next one.
Yes, sir. Sir, stratified.
2.11.
Yes, sir. Stratified cuboidal epithelium, excretory duct in salivary gland, stain HND into 100 magnification, and the innermost layer stratified cuboidal epithelium. Then they have labeled connective tissue and a blood vessel with endothelium. On the right side, once again, they have labeled a blood vessel with endothelium and connective tissue, sir.
So this is the summary classification of epithelial tissue. Epithelial, simple squamous, cuboidal, columnar, pseudostratified, stratified, and urothelium. Now we are going into the glandular tissue.
Glands, one is simple, compound, branched, tubulo-acinar, acinar.
It is simple and compound, and both can be branched or unbranched. Then tubulo-acinar and acinar. So that's the major classification. And number 2, read this.
Sir, yes, sir.
Padima.
Figure 2.12, sir. Unbranched simple tubular exocrine glands, intestinal glands. A. Diagram of gland. B. Transverse section of large intestine. Stain, hematoxylin and eosin. Medium magnification, sir. Surface epithelium is marked, sir, in both the images. And then the secretory cells are marked in both the images, sir.
What is this whole portion known as?
Sir, mucosa, sir.
Super. So what are the portions of mucosa? What is this? What is this? And what is this?
Sir, it is the top one is the lining epithelium, sir.
Very good.
And then in the middle there is lamina propria, sir.
Super.
And then the basement membrane, sir.
Basement membrane. What is this?
This one is muscularis mucosa, sir.
And what is this portion?
This is the submucosal portion, sir.
Very good, ma. And what is this portion?
Sir, muscularis propria, sir.
Super. Okay. So this is that understanding is very important. Mucosa, submucosa, muscularis propria. Okay. Now you can see only the submucosa is having bigger blood vessels, isn't it? That's why the tumor cells has to cross the muscularis mucosa before calling it as carcinoma. And if you see them, these are simple glands. Okay, these are all simple glands. And how many lining epithelium for each gland? Single lining epithelium. Only one.
Yes,
it is very important. See in this picture, these are tubular glands, simple glands with a single lining epithelium. And you see here,
so 213 simple branched tubular exocrine gland, gastric glands. A. Diagram of gland. B. Transverse section of stomach. Stain, hematoxylin and eosin. Low magnification, sir. Here they are marked surface epithelium of both the images, and then the secretory cells, sir.
And one more addition, can you see this fried egg appearance? This
Yes, sir.
what can be these cells ma with abundant pink eosinophilic cytoplasm? These are the parietal cells, surface secretory epithelial cells. So the neck
Okay.
and the body has the parietal cells, and these parietal cells are situated only in the gastric fundus and body. They are not present in the cardia and antrum. You all know stomach. When you draw stomach, how will you draw? This is the esophagus, like this. This is the gastroesophageal junction, and up to here is the cardia, and this is the fundus, and this is the body, and this is the antrum. So there are four portions of the stomach. This is cardia, this is fundus, this is body, and this is antrum. Okay, and these parietal cells are seen only in the fundus and the body. So by looking at
Okay.
the gastric biopsy, you can see whether they are coming from the cardiac antrum or from the body and fundus. Okay, so these are parietal cells and no parietal cells in cardia and antrum.
Why coiled tubular exocrine glands, sweat
Mm,
glands,
mm,
A. Diagram of gland. B. Transverse and three-dimensional view of the coiled sweat gland.
mm,
Stain hematoxylin and eosin, medium magnification.
mm,
And excretory ducts are marked in both the images, and then
mm.
secretory cells are also marked.
You cannot differentiate at least these are all like tubular gland that is coiled, isn't it? So see, this is the same gland that is getting coiled, whereas this portion is the duct, and this portion is the glandular portion. Usually the glands will be like this and ducts will be seen separately in other organs. Whereas in sweat gland, it's a coiled tubular exocrine gland. You have to understand, and they are lined by cuboidal cells. And remember I told you the height is equal to the width. That's why they are cuboidal cells. They can be single lining epithelium, and when they become the duct, they will become stratified. So that you can see it very nicely. These are these are single epithelium here, whereas it is multi-layered here. So the secretory portion will have single cells, whereas the excretory portion or ductular portion will have uh, stratification. And one more thing you have to remember, they have not shown it here. And if you see these glands, if you see enlarge these glands, they will have two layers. This is the lumen and luminal epithelial cells. Okay, and I will draw the nuclei there. And then when they secrete, how do they come out? Somebody has to press them. So there is a flattened cells outside, and these cells have the features of epithelium and muscle. So that's why it is called myoepithelium. So this is a luminal epithelial cell and abluminal, away from the lumen, myoepithelial cells. The tumors can arise from the epithelium, from the myoepithelium, or mixture of epithelium and myoepithelium. That's why you have to know those three two components of the epithelium and myoepithelium.
Compound acinar exocrine gland, mammary gland. A. Diagram of the gland. B and C is mammary gland during lactation. Stain hematoxylin and eosin. B is low magnification. C is medium magnification. Excretory ducts are marked in both the images, and secretory acina is also marked.
So can you see the difference between the this top one and the bottom one, isn't it? Here the secretory and the ductular portion, they look alike, whereas here excretory is here, ductular portion, and the secretory portion is here. These are minor ducts and these are the major ducts, and these are all the acini. And when you see it in this one, normally they are arranged in a lobular pattern. That lobular pattern is very important. Wherever the conditions you see the lobular pattern, that is benign. When it is doing the infiltrative pattern, then it is malignant. So that lobular pattern of normal arrangement is very important. Super. And you all know breast is a modified Sweat gland or sebaceous gland.
So it's a compound tubulo-acinar exocrine gland, a salivary gland, and the A shows a diagram of the gland, and B shows a submandibular salivary gland. So A and B excretory duct have been mentioned, and secretory tubule glands and the secretory acinar glands.
What is this gland, ma?
Salivary gland.
What gland?
Exocrine glands.
What
Submandibular
exocrine gland? Submandibular. Remember, there are three major glands. One is parotid, three pairs. What is the other one, ma? Submandibular. And then sublingual.
Sublingual.
These are the three major salivary glands. After that, you have minor salivary gland. I told you parotid is serous gland and sublingual is mucinous gland.
Mucinous.
And submandibular is seromucinous. That's why you see, if you go to the high power, what are these cells, ma?
This is mucinous acinus.
Very good. This is mucinous. And what are these?
Serous acinus.
That's why this is a submandibular gland. Even if they don't tell whether it is parotid, sublingual or submandibular, we can identify based on histology. Okay, parotid is predominantly serous gland, sublingual is predominantly mucinous gland, and submandibular is seromucinous glands.
It's a compound tubulo-acinar exocrine gland, submaxillary salivary gland, sir. So number 1 shows a secretory acinar element. Number 2 is connective tissue, then excretory duct, mucinous cells, serous cells. And 6 is the same, sir, excretory duct, and 7 is secretory tubular element, and 8 is excretory ducts.
I hope you understand the difference between the acinar and ductal. These are all ductal, cuboidal epithelial cells. Okay, these are all ductal, cuboidal epithelial cells. Whereas these are all the acini. They are more like a columnar cell when it is mucin secreting, and they may be cuboidal with eosinophilic cytoplasm when it is serous cell.
At 2.18, we have a figure of endocrine gland, pancreatic islet. A is the diagram of pancreatic islet. B is high magnification of endocrine and exocrine pancreas. Stain is hematoxylin and eosin, high magnification. The figure 2.19, endocrine and exocrine pancreas. Stain is Mallory Azan. Coming to the labelings, first is excretory ducts, and then they have labeled the blood vessels, and number 3 is exocrine pancreas, number 4 connective tissue capsule, number 5 endocrine pancreas.
Now we will cover pancreas. Pancreas and adrenal, they are 2 in 1 organ. Pancreas is having exocrine and endocrine, and adrenal is having cortical and medullary. So these are all the 2 organs that will have buy one get one free. And this excretory, the pancreas, you see the abundant granular cytoplasm. These are more like parotid glands. If you don't see the islets, you will miss the pancreas as a parotid gland, because they, depending upon the anatomic location, we call it different name. These are nothing but, pancreas is nothing but parotid gland plus islet cells, and these islet cells will look like small round clusters with eosinophilic to clear cytoplasm, and they will have lot of blood vessels. Remember, these neuroendocrine cells, they don't have ducts to secrete. Whatever the cell secreted here, for example insulin, glucagon and all, it has to reach the body, but they don't have the ducts. So otherwise the blood vessels are the ducts, they secrete them directly into the blood vessels. That's why all endocrine organs are highly vascular. Remember, in this way, they are not glands without ducts. The blood vessels acts like ducts in them. Do you understand that, Merlin? Can you repeat that?
Yes, sir. So the neuroendocrine cells that are present in pancreas and all other endocrine glands, they don't have a duct, but the blood vessels in them act as ducts. Therefore, they are all highly vascular because their secretions go directly into the blood vessels.
Yes. This is very important because when you do FNAC of thyroid, it will be hemorrhagic because blood will come immediately. That's why you have to remember all endocrine organs. When you do FNAC, it will be hemorrhagic.